The next part is autosomes and the sex chromosomes. So autosomes are those chromosomes which are in simple terms is the normal chromosome. Okay, like humans we have 23 pairs, no, 23 pairs. So 22 pairs will be your autosome. Okay, all those chromosomes for your, your liver, your kidneys, your lungs, your brain, etc., etc. Then 22 plus one pair of not autosomes but allosome. Okay, allosome or sex chromosomes here. So here autosome you had your 22 pairs, and then the sex chromosome is one pair, no? So the another word for sex chromosome is allosome. I hope you can see what I've written. I think it's clear enough. Okay. So in a diploid cell, there are two types of chromosomes known as homologous chromosomes. Okay. Except for the sex chromosomes, they are non-homologous. Okay, the sex chromosomes are not homologous. So we have 23 pairs of homologous chromosomes. The human male has 44 non-sex chromosomes termed as autosome and one pair of heteromorphic or morphologically dissimilar sex chromosome, that is one X chromosome and one Y chromosome. Okay. And the female has 44 non-sex chromosomes, that's autosome, and one pair of homomorphic, that is similar looking sex chromosome designated as XX. So this is easy enough. I think it's clear. Now going directly into the morphology. It changes with the stage of the cell division, and the mitotic metaphase chromosome are the most suitable for studies on chromosome morphology. Okay, so just like you do practical, you do during the mitosis phases, no? That's why you choose the root tips or the buds, no? So because those are the dividing cells, okay, the mitotic cells. So you can see the chromosome clearly during those stages. So when you look under the microscope, you can see the chromatid, the chromonema, chromomere, centromere, secondary constriction, telomere, and the cytolites. So let's look at them in detail. If you look at the structure, we can divide the chromosome into three parts: the pellicle, the matrix, and the chromonemata. Okay. So if this is the chromosome, no, you can divide it into three parts. So let us look what is that. So first one, we will have the outermost covering. Okay, just like a cell has remember a cell, if a plant cell, then cell wall and cell membrane, no? Even the chromosome, they have some sort of a covering. Okay, 1 minute, let me scroll. So the outer envelope around the substance of chromosome, it is a very thin and is formed of achromatic substances. Okay, so means these are not a part of the DNA material, but they are non-DNA. Okay, but there are some scientists which do not which deny its presence. Okay, they say that it's not true, it doesn't exist. So after the pellicle or the outer envelope, we would go to the matrix. So the matrix is the main part, that is which contain the chromonemata, but this is also formed of non-genic materials. Okay, it is the ground substance of chromosome. How do I say? It's something in which the DNA will float in, something like that. Okay, here the matrix will contain the chromonemata. What is a chromonemata? We will see again later. Okay, if I twist, I think there you can see, no? Look at the structure. Okay, so when you were a student in school, you used to draw chromosome like this, no? With one big bulb in between, or like this, like a ribbon or something. No, you used to draw like this, then like this, no? And then you used to give some line like this. This is all wrong. Okay, all wrong. Okay, what is happening is that when you used to draw that circle, no, and used to say that is the centromere, it is actually, centromere is actually a constriction. Means, suppose this is your muffler, and then you squeeze it from here. You squeeze, no, with your fingers, your muffler becomes like this. Or shawl or scarf, whatever you squeeze, it will be a constrict like that. You get it? And then here it will attach to those, spindle fibers. Na? So, if you draw this way, this is wrong, okay? How you used to draw in school, it was wrong because they try to make it simple. No, when you studied in school, they try to make it simple. But the thing is, the centromere or the centrosome or the kinetochore, these are the three names of the centromere. It is a constriction. Look here. Okay, there. Suppose you want to draw it longwise, you will be drawing it like this. Do you notice that I've given a constriction? This one, okay. Suppose that is your centromere, or another name is kinetochore or centrosome. So when you draw chromosomes, you will be drawing like this, okay, with a constriction in between. I think it's understood. Then other than the centromere, there are other constrictions also. Here there are some secondary constrictions. Then there is a satellite. This is with spindle fibers attached. These are the chromanemata, the matrix, remember the ground substance. And then the pellicle, which is the covering. Okay. Okay, let's move on. It's too big. Okay, what is the chromanemata? Inside the matrix. Remember, first we had pellicle, then we having matrix. So inside the matrix are two identical spirally coiled threads. So remember, just like you used to draw the DNA, no? When, like in the school, you used to draw like this, no? Yeah, this one only. It's a spirally coiled threads, a chromanemata. The two chromanemata are tightly coiled together that they appear as a single thread of about 800 angstrom thickness. It consists of 8 microfibrils in the form of a double helix of DNA. Okay, so the chromanemata is what we draw the DNA. Okay, then we go to the centromere, no? Chromomeres, or not centromere, chromomeres, they are in the form of small dense masses that you can see on the chromanemata. Okay, let's look at the image. I don't see it. Okay. These are more distinct in the prophase stage where the chromanemata are less coiled and more clearly visible during leptotene and zygotene stages of the meiotic prophase. There. Do you see those black dots here? These are your chromomeres, okay? If we draw it in simple terms, like this, no, suppose, like this, like black dots. Okay, during the leptotene stage, you used to see like that, black dots. Okay, I'm drawing like super magnified, no, using my finger, but there are some like this there, black dots. Something like that. Those are your chromomeres, okay? They take a darker stain because they are, they stain a little bit differently. Okay, because you can really see them during the leptotene and zygotene stages because they are less coiled. They will be lightly stained because they are less coiled. The rest of the DNA is tightly coiled, but in this chromomeres, they are loosely coiled, so they'll take light stain. Okay. Let's read what they have written here. While pairing during this one, I'm reading this one. While pairing during zygotene of meiotic prophase, the homologous chromosome pair chromomere to chromomere. Okay, so that's why it is a constriction, because of the less tightly coiled DNA. All right, chromomeres are regions of tightly folded DNA and are believed to correspond to the units of genetic function. How do I remove this thing? Oh, there. In the chromosomes. So when they pair, when the pairing happens, that is in the zygotine stage, they pair chromomere to chromomere, means the scam, spam, whatever. So they pair in the thinly coiled section to the thinly coiled section. Okay, so they pair with each other. Now looking at the chromatid. So during metaphase, each chromosome will have two symmetrical structure called chromatids. Each chromatid will have a single DNA molecule, okay? And both chromatids are attached to each other by the centromere and become separated at the beginning of anaphase, when the sister chromatids of a chromosome migrate to the opposite poles. So chromatid is nothing but your homologous chromosomes, okay? Means this one is one chromatid, this one is another chromatid, okay? So, and they align together from here, centromere to centromere. No, these two align together, the homologous chromosomes. Then, looking at the centromere, it is permanent. It is a small structure in the chromanemata and is marked by a constriction. Okay, so as I told you, when like we used to draw like this, no, like a ribbon or something, or used to make it black or something. Some kids used to do that. Something used to do like this, no, but it's not. It's just a constriction, just a constriction like this. Okay. It is known as your primary constriction or centromere or kinetochore. Okay, these are the three names. It has a constant position. Okay, and we use it to identify which chromosome this is. Let's say if this is chromosome 21, no, we know just by looking at the position. We know this one is chromosome 21. Let's say, for example, this one is chromosome 11. Okay, just by looking, if 1 minute, just by the position of the chromomere, okay, we will know. I mean, the centromere, we will know that this will be chromosome 11. Then so on and so forth. No, because this position is constant. Something like this, suppose. Okay. Then, for example, this one is chromosome, 12 or any other chromosome. So they have their own unique positions. So in the electron microscope, we see that the centromere, it is trilaminar structure. Okay, so 3 layer, 10 millimeter thick dense outer proteinaceous layer, a middle layer of low density, and a dense inner layer bounded to the centromere. So it's obviously it's made up of protein only. It has a thick dense outer, a middle low density, and a dense inner. Okay, so it's thick from thick. Oh, so something like this: thick outside, thick inside, and thin middle. Okay, thin density. So then they attach to the spindle fiber at this region. Obviously, you already know it's the centromere that attached to the spindle fiber during cell division. So the part of the chromosome which lies on either side of the centromere will be its arms. Let's say if this is chromosome 1, okay, so this will be the P arm and this will be the Q arm. Okay, then if suppose this is chromosome 12 or 12, this is the P arm and this is the Q arm. Okay, so wherever the position of the centromere is, the one on the top will be the P and this one on down will be the Q arms. Okay, So depending on the number of centromeres, the chromosome may be monocentric with one centromere, dicentric when there are two centromeres, polycentric more than two centromeres. Okay. Sometimes acentric without any centromere, and then diffused or non-located with indistinct chromosome. Okay. So we can divide them as chromosomes into certain categories based on the location of the centromere. Telocentric, they will be rod shaped. See here, the A, number A, this one. Telocentric will be like this rod shape. Means this one, no, one centromere is there. They are drawing the old-fashioned way. It's okay, easy to understand. No, they are drawing the centromere the old-fashioned way because it's difficult to draw in the constriction way. No, so it be like this. Okay, then it looks like a L or I shape. No, I think like a rod shape. So that is at the end. At the end, it will have just one arm. Then acrocentric, it is subterminal, means if these are the homologous chromosomes, okay, this is the main long arm, but here are the short arms. Okay, these are the constriction. I'm using my finger, so I'm not able to draw properly, so you understand it will be somewhere here, not exactly at the tip the way it was for the telocentric, but a little bit near the base. Okay, then you will have submetacentric, slightly away from the midpoint. Okay, so something like this, just slightly with the midpoint, and then the one with the center one will be the metacentric. Okay, these are your V-shaped chromosomes. The centromere will lie exactly in the middle of the two arms, and the two arms will be almost equal. So it is the centromere that controls the direction, that is orientation and the movement of the chromosome on the spindle. It is a point where the force is exerted when the chromosomes move apart during anaphase. Okay, so I'll stop here for today.